Welcome to the third part of our conversation on microbial genetics. For this video, we are going to talk about translation and just kind of give a big overview of what this process is about. So remember, we've been talking about gene expression, which is the process of starting with a molecule of DNA, using one of its strands to create a single-stranded molecule of RNA, which that process is called transcription. And then now we're going to use that piece of RNA, specifically the messenger RNA, and use it to create a protein. Now, this process is going to be called translation because it actually requires us to change languages. So, excuse me, we're starting with a molecule of DNA, which is made up of nucleotides, right? And then we made a molecule of RNA, which was also made up of nucleotides, and that was the process of transcription. Now that we have a molecule of RNA, again, the RNA is made up of nucleotides, but we're going to be making a protein, which is made up of amino acids. So we've now changed the language from nucleotides to amino acids. And that's going to require a translation device, which you're going to see on the next slide. So how are we going to do this? We are going to need three key players. We're going to need the messenger RNA to serve as the directions, because remember, the messenger RNA is anti-parallel and complementary to one of the strands of the DNA, right? So the messenger RNA still holds that important information that DNA had. But we needed this RNA because it is what the ribosome understands, and the ribosome is the protein-making machine, okay? So we're going to have the messenger RNA, which is going to be the directions. We're going to have the ribosome, which is the specific location where translation is going to happen. So think of the ribosome almost like the factory. That's the building where the amino acids are going to be linked together to build a protein. But so far, we have directions and we have the building. But what about the little worker that goes back and forth and grabs what's needed and actually puts the protein together? Well, that's going to be the tRNA, and that T stands for transfer. And we call it a transfer RNA because it is grabbing amino acids from within the cell and transferring it to the ribosome, where those amino acids will be linked together to form a big protein. So that means we're going to have three key players. So kind of big picture, and we're going to visualize this on the next slide. Messenger RNA says, I need the amino acid called methionine first. And one of the transfer RNAs goes out to the cell, grabs methionine, brings it to the ribosome. Then the messenger RNA says, okay, I need serine second. A transfer RNA goes out, grabs the serine, brings it to the ribosome, links the methionine and serine together, and we're started. Then methionine might say, hey, I need leucine third. Transfer RNA goes and grabs the leucine, brings it to the ribosome, it gets connected to the previous two. Okay, so this is kind of going to be big picture how these three key players interact. But before we move on, let's think about where inside the cell is this going to happen? Like which kind of area of the cell that we've talked about previously is this going to happen? Well, to answer that, you need to think about where are the ribosomes, because the ribosomes are one of our key players. And so in a prokaryotic cell, the ribosome, and really everything else on the screen, is found in the cytoplasm, because remember, there's no internal organization in the prokaryote. So what about in a eukaryote? Well, remember, Ribosomes are partially, or some of them are associated with the rough ER, but there are others just kind of hanging out in other areas of the cytoplasm because the cell needs so many proteins that there's just ribosomes all inside the cell. So we say that the location of translation for eukaryotes is also the cytoplasm. All right, let's go ahead and visualize this process of translation. So remember, we're changing languages. You can see the nucleotides that make up the RNA. That's going to be the A, U, G, C, C, A. Okay, those are the nucleotides. Now we need to write out the amino acid sequence. And so we're going to use this codon wheel on the right to figure out which amino acid is needed. Now notice it's called the codon wheel, and that's because in order to translate the messenger RNA, we are going to have to divide the strand of RNA nucleotides into 3. Okay, so we are going to start at the beginning of our strand of messenger RNA and divide them, like I just said, into 3, and we call those codons. So there we go. And when I write RNA, I just naturally tend to chunk it into 3s because that's what we're going to need. So there's our codons. So each codon is going to code for a specific protein. And how you figure out, or I'm sorry, let me rephrase that. Each codon codes for a specific amino acid. And then all the amino acids will link together to make the protein. So AUG is always going to come first. We call that the start codon. So remember, we need to think of a few things with messenger RNA. It always runs 5' to 3'. It's going to include the base uracil, and it's going to start with AUG. Always, at least for our cases, okay? So how do we figure out which amino acid that is? Well, you're going to start in the middle of the codon wheel. So right in here. So then you're going to work your way outward. So we're going to start with A. We're going to work our way out to U. Make sure you're watching the screen to see where my mouse is going. And then you're going to work your way out even more to G. So notice that leaves us to MET, which is MET. That stands for methionine, but all you will need to write down is the MET, okay? So hold on, let me get you there, right there, okay? So our start codon codes for the amino acid methionine. Notice I went ahead and I numbered the remaining codons. So codon 2 is CCA. So again, you're going to start in the middle at that C. You're going to work your way out to the next C, and then work your way out to the A. And notice that gives pro, which stands for proline. All right, so now we're going to do this again. The third codon is GUA. That's going to be G is right here. Work our way out to U. Work our way out to A, and that's going to give us valine. So on your own, go ahead and figure out what the remaining, how about just the next 2 codons give us. So I'm going to move a little faster, but feel free to pause the video. So GAA is going to give us GLU, which is glutamine, and then UCC is going to give us SER, which is serine. Now, notice I don't have a blue arrow coming down from the final codon, which is called the stop codon, and that's UGA. So if you follow out UGA, notice it's a little circle, and there's a little key at the bottom of the codon wheel that tells you that's a stop codon. That means the process of translation is done. Everything needs to stop. There's no more amino acids that need to be added. So think about this. So far, the amino acids are methionine, proline, valine, glutamine, serine. Do you really think there's going to be an amino acid with a simple name like stop? No, right? It's not that easy. So what that means is we're just done. You don't write anything there. Serine is the last amino acid, so we're done. Okay? So essentially, kind of big take home right now is our methionine, the messenger RNA is going to be divided into groups of 3 nucleotides. Each group of 3 RNA nucleotides is called a codon. So the messenger RNA starts with a start codon. It ends with a stop codon. We use this codon wheel to figure out what each codon codes for. And you start in the middle, work your way out, and that's how you identify the amino acids. So we've read our directions and built our protein. On the next video, we're going to talk more about the details of how these all interact. Let me know if you have any questions.